I think one of the most fascinating things about metabolism is it's involved in every disease. At the basis, metabolism is the thing that makes cells, that make tissues, that make organs work.
Metabolism is embedded all across the Radcliffe Department of Medicine to come up with biological insights that benefit human populations.
One of the challenges we have in our approach to disease is that I think we've looked too narrowly in the past. And one of the most exciting opportunities of the moment is to combine the insights from colleagues across the RDM.
This network will cover both basic and clinical research in aspects of hematopoiesis, immunology, diabetes, liver disorders, and cardiovascular disease.
As amazing as my research institute is, I do need certain techniques or models or tissue approaches that I can't do by myself. And in that sense, first I have to know that they exist within RDM, which is where the cross-cutting theme comes into place.
Many of the metabolic processes that are required for the normal functioning of the cell are highly iron dependent. And so we wonder what happens when iron is lacking.
We have identified that a highly conserved protein called MPRL3 is able to regulate a central metabolic controller. It's not just that not enough iron means not enough hemoglobin, which means anemia, but also iron acts as a metabolic signal.
We've been troubled by an unusual feature of diabetes. While it's quite clear that hyperglycemia drives disease, treating the hyperglycemia does not remove the complications of cardiovascular disease. And that paradox really drew us in about 10 years ago to trying to unpick the mechanisms by which glucose fundamentally reprograms cells such that they have a memory of prior exposure. And that memory is what we're trying to target therapeutically.
Detecting metabolism with normal MRI is quite tricky because of the low concentration of metabolites. We have a special process called hyperpolarization, whereby we take a sugar, we label it with carbon-13, which is non-radioactive, and then we hyperpolarize. This transiently gives us a boost of signal to this sugar. So we take the injection that we form into the MRI scanner, inject it into our participant, and start imaging metabolism in real time.
Metabolism changes in disease, and so being able to detect that means that you can get interesting information very, very rapidly. You can do it multiple times over the course of a disease, so as the disease changes or as the patient's receiving treatment and that response to treatment.
So I started off very interested in metabolism and the hormonal control of metabolism. And it became very clear that things are changing very strongly through the day. So a meal you have for breakfast works differently in your body than a meal you have at dinner time. The thing I'm most committed to is driving all of the major insights in circadian biology and circadian medicine, reduce them down into interventions that we can apply to human populations.
It will speed up the research, it will enrich the research, make it more diverse, help other people working on similar areas to do their work faster.
We want to be able to get this out to a wider audience. We're trying to expand it and drive it into various clinical domains, looking at neurodegenerative diseases, looking at cardiovascular diseases, really opening up the span so that it's available to more patients.
By working with collaborators across the Radcliffe Department of Medicine, we're able to draw in expertise, technologies, analytics that allow us to open up really new channels of investigation.